Imagine typing just a single sentence into a prompt, like, cure this novel drug-resistant respiratory virus.
Right.
And in the time it takes you to drink your morning coffee, an AI doesn't just guess a chemical structure. It hallucinates a perfectly optimized molecule.
Yeah.
And then it mathematically proves its safety and mechanism to the FDA and autonomously simulates a 10,000-person clinical trial to verify it works.
I mean, it sounds like science fiction, truly. But the foundational architecture to actually execute that end-to-end process is, well, it's no longer theoretical.
Wow.
Yeah, it is being assembled right now in the field of computational biomedicine.
Welcome to the Deep Dive. So our mission today is to explore a stack of incredibly dense, cutting-edge AI research papers to answer one mind-bending question. How does an interconnected web of artificial intelligence actually do this?
Right, because we are looking at a paradigm shift here.
Exactly. We are transitioning away from the era of massive black box large language models, the ones that are essentially just highly advanced autocompletes guessing the next word.
Yeah, the old way of doing things.
Right. And we're moving into an era of verifiable neurosymbolic architectures. So for you listening, we are tracing the journey of a single hypothetical cure from your human prompt all the way to a phase 3 trial.
And that journey requires a fundamental shift in how we build AI, because a standard neural network, it just cannot invent a novel drug.
Right.
It lacks the capacity to actually understand biology. I mean, if you just feed an AI millions of medical textbooks, it doesn't form a conceptual understanding of a virus.
It just maps the words, right?
Exactly. It just maps the statistical frequency of words associated with a virus.
Okay, let's unpack this. Because the very first massive hurdle here is moving from that statistical guessing to genuine conceptual intelligence.
Yes.
To invent a drug, the AI has to grasp abstract biological concepts without getting completely, overwhelmed by the chaotic volume of sensory data.
Which brings us to a revolutionary framework detailed in the research called CATES-NET.
CATES-NET.
Right. It stands for Concept Abstraction and Task-Solving Network. And the core problem CATES-NET solves is decoupled thinking.
Decoupled thinking. Okay, what does that look like?
Well, in humans, if I say the word dinner, you instantly evoke a conceptual understanding of dining. You don't need to be actively smelling roast chicken or looking at a plate to understand what dinner is.
Right, I just get the concept, the vibe.
Exactly. Your brain has compressed thousands of high-dimensional sensory experiences, the smells, tastes, sights, into a tight, low-dimensional concept.
So the thought is entirely decoupled from the direct physical perception.
Yes. And CATES-NET brings this exact cognitive structure to AI through a dual-module framework.
Okay.
First, you have the concept abstraction, or CA module. It acts like a funnel.
A funnel for data.
Right. It takes massive high-dimensional sensor motor data, like the endless complexity of cellular structures, and compresses it into a highly constrained 20-dimensional concept space.
Oh, wow. So it just forces out all the noise.
It forcefully strips out the noise. Yes. Then, the task-solving, or TS module, uses those compressed concepts as gating signals to make judgments.
Gating signals.
Yeah.
The research actually highlights a fascinating breakthrough experiment on this, using the CFAR 100 dataset to prove how this conceptualization changes the game.
Oh, the learning by communication experiment? Yes.
Yeah. So they set up this scenario where they had a teacher network that learned 100 different visual concepts and a totally separate student network that only learned 99.
Right.
The category of apple was deliberately withheld from the student's training data.
So the student had absolutely no mathematical representation of an apple whatsoever.
None. But the teacher was able to successfully transfer the knowledge of an apple to the student. And it didn't do this by, sending thousands of gigabytes of image data or forcing the student to relearn its entire data set.
Yeah.
It worked purely by aligning their internal semantic spaces.
They engaged in what the researchers call conceptual communication.
Right. So instead of forcing one giant AI to read and memorize the entire medical library, this is like delegating to a team of highly specialized experts who can pass a perfectly condensed brief to each other.
That analogy is close, but the reality is actually much more alien than human delegation.
Wait, really? How so?
Well, human experts still have to translate their concepts into clumsy, imprecise words to communicate, right?
Yeah, it's true.
These independent AI agents are bypassing language entirely. They are passing pure, raw, 20-dimensional mathematical concepts to one another directly.
That's wild.
What's fascinating here is the scalability this introduces, because in traditional deep learning, knowledge is entangled across billions of parameters.
Right.
So teaching an AI something new often means re-updating the whole monolithic system. CatsNet bypasses that completely. It allows for scalable, decentralized, multi-agent collective intelligence.
And the research noted that when they mapped the activity of this AI model, the concept abstraction module actually aligned perfectly with the human brain's semantic control network.
Yes.
So we are essentially engineering artificial systems that structurally mirror human conceptual intelligence.
We are. And honestly, that is the only way a multi-agent system can handle the next phase of your prompt.
Okay. The next phase.
Right. Because once these specialized agents share their conceptual understanding of our target virus, they are staring down a sea of biological variables.
Right, because biology is infinitely messy.
Infinitely.
Like if the agents are trying to predict, say, a specific cardiotoxicity trait, basically making sure our new drug cures the virus but doesn't accidentally, destroy the human heart, they have millions of biological features to consider.
Millions.
So how do they pick the exact chemical targets without wasting decades computing every single possibility?
Well, they have to overcome what's known as the feature selection bottleneck. Historically, to find the most important features in a massive data set, computer scientists used a brute force method called greedy forward selection.
Greedy forward selection.
Yeah. You test one biological feature, see if it improved the model, then test the next, and so on.
That sounds exhausting.
It is, because to find the absolute best combination, you have to train a massive number of separate models. Mathematically, it requires big O of K times D models.
Okay. And in English?
In modern biology, where you have millions of features, the compute time required to do that would literally outlast the universe.
Outlast the universe. Okay, so that's a problem.
A huge problem.
But the research introduces a highly elegant workaround called sequential attention. And the mechanics of this are just brilliant.
Yes.
It completely bypasses the need to train millions of separate models by using a global trainable softmax mask alongside something called the Hadamard product.
Right. And the Hadamard product is simply an operation that performs element-wise multiplication.
Okay.
To visualize the impact of this, imagine a master chef trying to perfect a complex soup.
Okay, I like food analogies. I'm listening.
The old method, greedy forward selection, would be like the chef tasting every single ingredient in a massive kitchen, one by one.
And making 10,000 different pots of soup to find the right combination.
Exactly.
Yeah.
Which takes forever.
Yeah.
But sequential attention, using the Hadamard product and the softmax mask, allows the AI chef to essentially take one single sip of the broth and instantly, fractionally weigh all 10,000 possible spices in the kitchen at the exact same time.
In one sip.
In one sip. The softmax mask scales down the mathematically irrelevant features, allowing the network to automatically hone in on the most critical biological variables in a single pass.
And the researchers proved that this incredibly fast single-pass method mathematically matches the strict guarantees of a classical algorithm known as orthogonal matching pursuit, or OMP.
Right.
So you have rigorous mathematical certainty, but at a fraction of the computational cost.
Exactly. It allows the multi-agent system to instantly find the precise biological target for the cure. But that assumes we have a massive historical data set to analyze.
Oh, which brings us to a massive twist in our scenario. What if our prompt is asking the AI to cure a brand new, novel pathogen? By definition, there is no massive data set of historical variables for sequential attention to filter through.
Right. There is no soup to taste.
No soup. So what happens?
This is where the architecture utilizes polymath learning. It pushes the system into a realm of extreme data efficiency.
Extreme data efficiency.
Yeah. When you are dealing with novel pathogens or rare orphan diseases where massive data sets simply do not exist, polymath learning allows the AI to use one-shot reinforcement learning.
And it does this by using a single metasample.
Yes.
Now, I have to stop you there because when I read this in the research, it sounded a bit like magic.
It does sound like magic.
The researchers strategically engineer one single training sample that combines physics, chemistry, and biology. But how does one single sample teach an AI the fundamental rules of the universe?
It's a completely fair question, and it requires shifting how we view training data. We are used to thinking of data as historical examples.
Like showing a computer 10,000 pictures of a cat.
Exactly, but a metasample isn't a historical record. It is a mathematically dense construct that embeds physical invariance.
Okay.
Think of it like a unified equation. It forces the reinforcement learning agent to balance energy conservation, molecular binding affinities, and cellular dynamics simultaneously in one highly constrained environment.
So what does this all mean? If we're looking for a cure to a brand new virus, polymath learning acts as a reasoning synthesizer rather than just a probability amplifier.
That is the critical distinction.
Okay.
A standard LLM amplifies the probabilities of patterns it has seen millions of times. A reasoning synthesizer understands why molecules interact based on the underlying laws of physics.
Ah.
Because the agent understands the fundamental constraints of thermodynamics from that metasample, it can generalize its knowledge to entirely new molecular modalities. It solves the data scarcity problem that normally paralyzes rare disease research.
Okay, let's look at where our single prompt has gotten us so far. Our multi-agent AI team has conceptually communicated the goal of curing the virus. It used sequential attention to zero in on the highly efficient chemical targets, and polymath learning to design a totally novel molecule that obeys the laws of physics, even with zero historical data. It is a stunning scientific achievement.
But there's always a but.
Here's the reality check. The FDA does not approve black box guesses.
They absolutely do not. Regulatory bodies require a verifiable mechanism of action. You have to prove exactly how this novel drug works in the human body, step by step, with causal proof.
Which makes sense.
Of course.
Yeah.
And this is where standard large language models completely fall apart.
Because they hallucinate.
Right.
The research points out that a general LLM, even a massive one with 32 billion parameters, has a factuality score of only around 40.2%.
That's failing.
Miserably. Its validity score for adhering strictly to medical ontologies is just 43%. If you hand a 43% accurate document to the FDA and say, "Trust us, the computer made this," you will be laughed out of the room.
Or arrested.
Yeah, seriously. You cannot have an AI hallucinate a chemical reaction inside a human liver.
Which is why the architecture shifts away from general LLMs at this stage and deploys a model called GraphMERT.
GraphMERT.
Yeah. And what is staggering about GraphMERT is that it is a compact encoder model. Only 80 million parameters compared to 32 billion.
Huge difference.
Yet it achieves a FACS score of nearly 70%.
Wait, how does a model that is a fraction of the size become nearly twice as factual?
Through a rigorous process called neurosymbolic distillation.
Neurosymbolic distillation.
Right. GraphMERT doesn't predict words based on internet text. It uses masked language modeling and masked node modeling to take unstructured biomedical text and turn it into reliable symbolic knowledge graphs.
Okay, let's clarify what masking actually means in this context, because it's vital to understanding why this works.
Sure. Imagine a massive multidimensional web mapping every known disease, protein, and chemical compound.
Okay. Got the web in my head.
Masking means the AI deliberately hides or masks a specific connection in that web.
So it creates a blind spot.
Exactly. It essentially blindfolds itself to a known biological fact and then forces the system to deduce what that missing link must be based entirely on the surrounding causal neighborhood.
Oh, that's smart.
Yeah, and by doing this millions of times, it builds a rigid, unshakable map of relationships.
It's inferring the underlying rules of the biology rather than just memorizing it.
Precisely.
Here's where it gets really interesting. Because GraphMERT creates these rigid maps, it provides sentence-level provenance for every extracted fact.
Yeah.
It's the difference between a student guessing the answer on a test based on the vibes of the question and a student bringing peer-reviewed citations for every single sentence they write. It anchors the AI.
And that anchoring is the only way to get to clinical trials. GraphMERT supplies the human regulators with transparent, auditable proof.
Right.
It provides the exact symbolic tracking required to prove that the novel molecule invented by the Polymath agent obeys peer-reviewed biological axioms.
Which brings us to the ultimate bottleneck in the entire pharmaceutical pipeline.
The trials.
Yeah. The AI has designed a drug that is mathematically verified and biologically sound. The FDA has reviewed the mechanism of action. But now, you have to test it in phase 3 clinical trials.
The graveyard of drug development.
Exactly. And listeners who follow computational biology know that phase 3 trials are agonizingly complex.
Oh, yeah.
We aren't just talking about giving a pill to 10 people. We are tracking deep, longitudinal electronic health records for thousands of patients across multiple years.
Right.
In standard neural networks, this creates a massive technical hurdle called information dilution.
Yeah, if you process longer and longer sequences of patient data using standard deep learning, the earlier information naturally degrades.
It's like trying to remember the exact wording of a sentence someone said at the beginning of a 3-hour meeting. By
Exactly.
the end, the details are a blur.
To solve this and automate the trial design, the architecture deploys specialized clinical frameworks like AutoTrial and PyTrial.
Auto trial and PyTrial.
Right. AutoTrial is remarkably effective. The data shows it boasts a 60% win rate against GPT 3.5 for generating trial criteria.
Wow.
It processes over 70,000 precedent trials and uses GraphMert's neurosymbolic grounding to generate eligibility criteria that strictly adhere to established medical ontologies, like the Unified Medical Language System.
But PyTrial is the framework that actually fixes the information dilution problem we just mentioned, using a mechanism called attention residuals, or ATNRs.
Right. Add-and-rest replaces simple additive residual connections with a learned softmax attention mechanism.
Okay, and practically speaking.
What this does practically is ensure the network can track deep, long-form clinical events over years without losing the signal of past anomalies.
Oh, I see.
It remembers that a patient had a highly specific slight enzyme spike 6 months ago and accurately correlates it with a new symptom today.
And if you are dealing with a cure for an orphan disease, disease so rare that you literally cannot find enough humans on Earth to run a traditional phase 3 trial, the architecture brings Polymath learning back into the fold.
Yes, it does.
It synthesizes highly realistic, scientifically grounded virtual patient records to simulate the trial first. Think about how wild that is for you.
It's incredible.
If you've ever waited for a breakthrough treatment for a loved one, you know the phase 3 trial is an agonizing, years-long waiting period. This architecture is designed to simulate that waiting period away.
This raises an important question, though.
Okay.
When you simulate a trial or monitor a real one, you are constantly scanning for adverse events. You're looking at MRIs and ECGs.
Right.
How does the AI flag an anomaly without reverting to a black box? This is where GraphMert's neurosymbolic knowledge graphs are critical. If the AI flags a lesion on an MRI, it doesn't just output anomaly detected.
Because that wouldn't be enough for the FDA.
Exactly. Because it is anchored in medical ontologies, it can explicitly explain why that anomaly is relevant based on established clinical guidelines. It provides peer-reviewed justification to the human scientists monitoring the trial.
So it is an unbroken chain of logic from the first conceptual thought all the way to the human MRI scan.
Yes.
KEDSnet divides the labor into specialized agents that communicate conceptually. Sequential attention parses millions of variables instantly. Polymath learning designs a totally novel molecule using a thermodynamic metasample. GraphMert proves the causal science to the FDA. And AutoTrial and PyTrial design and execute the long-form human trials.
It is a breathtaking end-to-end pipeline.
It really is.
But the research papers we reviewed push this one step further.
Wait, further? How?
Because we have to ask, what happens when this entire brilliant multi-agent system hits a wall? What happens when the AI encounters a biological roadblock that it fundamentally has not been programmed to solve?
Oh, this is the absolute apex of the research.
Yes.
When the AI hits the limits of current human knowledge, it utilizes a framework called the Darwin Gödel Machine, or DGM.
The DGM is a framework for open-ended autonomous self-improvement. It relies on a concept called symbiotic self-evolution.
Symbiotic self-evolution. So, the AI multi-agent team operating under cooperative frameworks like Dynamate enters a self-reinforcing cycle.
Right.
When they get stuck, they don't wait for a human programmer to intervene and update their code. They generate their own synthetic polymath primes.
Exactly. They engineer their own highly complex, novel training data to forcefully induce the next leap in reasoning.
Hold on, you're losing me here.
Okay.
You're saying the AI writes its own synthetic polymath primes.
Yeah.
What does that actually look like in practice? Is it writing new software code to improve its processing, or is it literally making up new physics to solve the biological problem?
If we connect this to the bigger picture, it is doing both, intertwined.
Both.
Yeah.
Mm-hmm.
If the existing laws of molecular physics as currently mapped by human science aren't sufficient to find a chemical pathway to cure the disease, the Darwin Gödel Machine autonomously begins engineering new scientific axioms.
Unbelievable.
It rewrites its own source code to process entirely new branches of physics and chemistry that it invents specifically to overcome that exact roadblock.
It evolves its own intelligence to match the complexity of the disease.
Yes.
This is a staggering paradigm shift. It means the future of medical breakthroughs will no longer be artificially limited by human cognitive capacity. It replaces a century of manual, physical trial and error with verifiable, self-evolving neurosymbolic engineering.
It scales human intent without being bottlenecked by human comprehension.
But that exact scalability leaves us staring at an incredibly profound precipice to end on. We started this deep dive exploring how these systems use GraphMERT to rigorously prove their science to the FDA because we crave precision.
Right, we want transparency.
We want an unbroken chain of logic that fits neatly into human understanding. But if a self-evolving AI system like the Darwin Gödel Machine eventually has to invent entirely new branches of physics or formulate new biological axioms just to cure a novel disease,
Yeah.
will human regulators and scientists even be able to comprehend how the cure works? Or are we moving toward a future where we will simply have to blindly trust the medicine? Something for you to think about until next time.